Next one. 5. Use the information below to answer the following question. Oh, let me look at this first. So what does this look like? I don't know. It looks like a cube to me, but I have no idea what it is. So let me read. The student conducts an experiment at home to test the effect of different covers on the melting rate of ice. So different covers, I'm going to underline that, and melting rate of ice. I want to see how this is affecting that. So I'm going to say here, this is my cause, and this is my effect. So if you remember that, that was my independent variable, and this is my dependent variable. Remember, my independent is my cause, my effect is my dependent. All right. Yeah, and that must be something that the scientist changes. Yeah, they change the covers. All right. The student places identical ice cubes on separate trays of known mass. So they're all the same mass when they start. Okay, I get that. The student covers each tray as shown below. What's my question? The trays are placed on the same table. After 10 minutes, the student removes the covers, discards the excess water, and calculates the mass of each cube. Okay. So it's starting at a certain mass. So they all start with the same mass, and then mass end is basically after, you know, I let them sit there. Okay. Okay. And this is when I remove the cover. Which of these is the dependent variable in the experiment? Oh, I think I already know that. So if I look back up here, I know that I, as the scientist, changed the different covers to see how that impacted or affected the melting rate of ice. So I think I know what my dependent variable is. The melting rate of ice depends on the different covers. So it must be the melting rate of ice. Let's see if it's here. Shape of each cube. I don't see melting rate of ice. So maybe they're trying to trick me here. Let's see. Shape of each ice cube. I don't know. I don't think that'll change much. I don't think. Maybe that could be the mass. Well, I am measuring mass, and that is changing. Yeah, so if it melts more, it's going to have less mass. So yeah, that must be it. I think this one, I'm going to star that one because I think that's it. Yeah, no, we never talked about temperature. Material covering, no, that's my independent variable. They're trying to trick me there. So I really think it's B. This is it. Yup, this has to be it. It's the mass of each cube because I was measuring the mass. And that's what was changing. Yup, and it had to do with what I was doing. Exactly. 6. Which of these is the control in the experiment? Control, I know the control is the one that I did not change really. Okay, let's see. Let's look at these. This has plastic, foil, paper towel. Oh, I didn't change this one. Ice doesn't come with a cover, so I didn't change that one. Ice comes as normal, not covered. So I know that that must be my control, that I'm going to be comparing all of these guys to that one there. So let's see. Do I have that there? Mass of the trays. Nope, that's not it. Tray with no covering. That's the one. There we go. I know that's it. Let's see. Let me make sure. Yep. B is my answer. Okay, number 6, a lot of words. All right, let's look at this. So first I want to look at my graph, see if I can just save some time here. The effect of catalase, oh, I remember that ACE is my enzyme from my awesome biology teachers here at Northwestern High School, and the effect of catalase on hydrogen peroxide. Okay, so the oxygen released is there, and then I have time here, and I'm talking about some liver, which is nasty, and some potatoes, which I like. Okay, here's my liver. I'm going to put an L there. Here's my potato. Okay, which of these factors was not controlled in the experiment? I have no idea because I didn't read it, so let's read. Catalase is an enzyme found in plant and animal cells. I was right. Hydrogen peroxide is a harmful substance found in cells. Catalase causes hydrogen peroxide to break down into water and oxygen. So I'm going to say that catalase, catalase, there we go, basically means that the hydrogen peroxide, it basically breaks it down. So I'm going to do this, and I'm going to put a crossing out so I remember. A student conducted an experiment to determine whether plant and animal cells have the same amount of catalase. Okay, do animal and plant cells have the same amount? That's what I want to know. She used liver and potato cells. Liver must be my animal. And this must be my plant, the potato. Okay, she used liver and potato tissues in the experiment. The student followed the procedures. Label 2 identical test tubes. Okay. Pour 10 milliliters hydrogen peroxide solution, 1% concentration to each. Okay. So I have 2. They each get the hydrogen peroxide. Add a small piece of liver tissue to test 1. So I'm going to put my liver there. Potato goes here. Collect the oxygen released from each test tube for 25 seconds. Okay, so I'm going to say I want to see how much oxygen is coming out. I don't know. I want to figure that out. Measure and record the volume of oxygen. So yeah, I want to figure, okay, that looks good. So what was not controlled? Control I know means that I'm not changing it. The time for the oxygen release. No, each test tube gets 25 minutes, so that's controlled. Each of them gets the same thing. So I definitely controlled that. I didn't change it. The volume of hydrogen peroxide solution. Let's see. No, both of them get 10 milliliters. It says it right here. So I'm controlling that. That's not changing. Each of them gets the same. The volume of the test tubes. Let's see. The volume of the test tubes. Let's see. Test tube 1. Looks like it says 2 identical test tubes. So I don't know. But then I'm measuring volume, so I don't know. Let's see. D, the mass of the liver and potato tissues. So it says here, add a small piece of liver tissue, a small piece of potato tissue. This looks like this must be my answer. Alright, so I'm going to go with D. There we go. Alright, let's go to the next one. Number 9. Which of these should be done before beginning a laboratory investigation? Collect data. Collect data means that I'm getting information. How am I going to do that if I haven't done my experiment yet? No, that doesn't make sense. Review the procedure. That's not a bad idea. Yeah, I should probably do that before I do anything in the lab, right? Conclusions come at the end. If I think of a story, it comes at the end anyways, the conclusions. No, that doesn't make any sense. Record data on tables. Yeah, that doesn't make sense either. I need to do it before I get the data. No, I know that's B. That makes sense. 10. A student's hypothesis is that increased exercise caused increased heart rate. Let's see, let's think about that for a second. Yeah, that makes sense. If I exercise, my heart pounds. Heart rate is determined by taking the pulse, which is measured on beats per minute. Okay, taking the pulse, beats per minute. Which of the following data would support the hypothesis? That means that which one of these would say that the hypothesis is correct. So I'm going to look for one of these that says increased exercise causes increased heart rate. So increased exercise is kind of like running or doing something active. So let's see. When sitting, her pulse was 70. When standing, it was 50. So it was more while sitting than standing. That doesn't make sense. More exercise should be more heart rate. And here, wait. Yeah, standing. You're standing, that's more exercise? No, that doesn't make sense. Standing is more, that should be more. When running, her pulse was 100. When sitting, her pulse was 60. Okay, so sitting 60, running 100. This makes sense. Sitting is less than running. Yeah, and running is more. I'm going to stay with that one. Let's just make sure here. Sitting is 90, walking is 90. No, these are the same. This doesn't make sense. Running, 65, standing, 70. No, you should be more while running. Nope, that doesn't make sense. B is my answer.